Okay, I can do that part. That's the hard part. All right. Good morning. Good morning. Whew. I don't know why it's hard to get this to a little bit closer is better. You hear me in the back? Good morning, everybody. Although it's hard to get it to actually clip. We were in the middle of amino acids somewhere. Almost to the end, right? PKU, thank you. All right, terrific. So just a few more things, unique things about amino acids for us to think about. So probably you've heard of PKU, phenylketonuria. It's a genetically inherited disorder, or we call it an inborn error of metabolism. And the way we treat it is by giving phenylalanine restricted specialty formulas. And so infants will be given a formula that contains no phenylalanine mixed with a tiny bit of breast milk or regular formula to meet just enough, to provide just enough phenylalanine to promote growth. And as people, get older, and children, and even to adulthood, special formulas are part of their life. They can eat, they have to avoid regular high protein types of foods, and they can live quite a long and healthy, productive life with the way we manage people. So it's just something to think about. And the way, what the problem is, the inborn error is that this enzyme, phenylalanine monooxygenase, which I'm just going to point out to you that in all of my slides, if you look at them in color on your computers, I always put enzymes with a yellow box and purple writing. It's just a pet, habit of mine. This should help you as you're looking at reactions when we go through the urea cycle or whatever. I'm labeling enzymes that way, okay? So this enzyme, there is an inborn error in it, and it's defective. So phenylalanine, essential amino acid, right, gets degraded to a non-essential amino acid, tyrosine. So this is a normal way that we produce tyrosine. In these people, they can't do that, right? So they are not, tyrosine actually becomes conditionally essential in these people, babies, and then kids and adults. And then the other thing that happens is when this enzyme is not functional and you consume normal foods containing phenylalanine, you get phenylalanine and its byproduct toxic, builds up and becomes toxic, phenylpyruvate. So you get both high levels of serum phenylalanine and phenylpyruvate, and that's how we monitor how well someone's adhering to their dietary restrictions, is we monitor their serum phenylalanine and phenylpyruvate. Okay, what else? The other thing you need to know about amino acids is that in some proteins, after you make a protein, the amino acids, some of the residues in those newly synthesized proteins can be modified. We call it post-translational modification, right? Some of the things that you may have heard of is histidine can be 3-methylated. I'll click the other two as well. Hydroxyproline, hydroxylysine are just a few examples. Once they are modified, they can't be unmodified, okay? So once they're hydroxylated or they're methylated, they can't go back to their native state. And when that protein gets broken down, we see those metabolites in the urine, okay? So 3-methylhistidine, I'm hoping you're not learning that's, what do you know about 3-methylhistidine? Do you know anything about, have you ever heard of this amino acid? Anybody in the room? So, no? Okay, well that's good then. We used to use it as a marker of muscle mass, actually, because it is highly prevalent in a protein called actin, which is one of our big, muscle proteins. So, we had a correlation to height, and you could use it to calculate or estimate muscle mass. But what we also know now is that actin is in intestine, and it's in other protein and other tissues in the body, right? So it's not a specific measure of muscle mass, so hopefully you're not learning that, so that's wonderful. But you will see it in old textbooks talking about 3-methylhistidine. And then hydroxyproline and hydroxylysine, do you have any idea what protein might contain these? Have you ever, probably maybe in biochem, I don't know. These are very unique amino acids, these hydroxylated proline and lysine residues. No one in the room? Any idea? It's a protein that's really important for wound healing and keeping our tissues together. Collagen, right? So we talked about collagen. I'm going to show you a picture of the structure of collagen, but it's a very strong protein, right? And that's a good thing because that's important to keep our teeth in place and other things. Hydroxylation reactions in general, probably not always, but in this case, require a vitamin. So when you think about wound healing and collagen synthesis, you have any idea what vitamin you might guess? Take a wild stab. Vitamin C, right? Those of you in MNT, and you've had this in basic nutrition, right? Your teeth fall out if you've got a vitamin C. Scurvy, think about the scorbutic gums and all that. Capillaries get really fragile, right? That's about collagen. And you can't make it if you don't have adequate vitamin C, so if you're deficient. So the great thing about collagen, and this isn't the best, and it's a little it's a triple helix structure. So it's got 3 chains, and the whole thing is about the hydroxyproline. I didn't show, it doesn't have hydroxylysine but labeled, but it is in there. And the proline has a really strange structure as well, and it allows for this helical structure to form, and then when you get the quaternary and tertiary structure, it really forms a very strong triple helix, which is why it's so important for our structural integrity. So hopefully that'll make it easy to remember that. Last, I think, or almost last, about amino acids, that we use amino acids in the body for neurotransmitter synthesis, and tryptophan and tyrosine are particularly important. Serotonin being a neurotransmitter, and melatonin, norepinephrine and epinephrine. What class of hormones are epinephrine and norepinephrine in? Catecholamines, right? The stress hormones. So that's an important thing to think about. And then glutamate. Glutamate has such an interesting life, right? I've told you it's the intracellular hub of amino acid metabolism. And it also can act as a neurotransmitter itself, and it gets converted also to GABA, gamma-aminobutyric acid, in the brain, an excitatory neurotransmitter. So really an important amino acid, and it's not essential, right? We make it in a lot of different pathways that we're going to talk about. And then I'm going to just pose this question. There is a class of medication called MAO inhibitors, still being used, but not as much as it used to be, for treatment of depression. So some people don't do well on the serotonin reuptake inhibitors that are kind of more, they've taken over. But MAO inhibitors, especially in older folks that have been on this kind of medication for a number of years, will still be on it. So it's something that you may see. And as a dietitian or a dietetic technician, you might actually have to instruct someone to avoid foods high in tyramine. So let me tell you why that is. And I also have just a point here, and we'll show this in the next slide. But tyramine is not tyrosine. Make sure you just realize that. It's derived from tyrosine. So it's an amino acid derivative, but not one of the amino acids itself. So here's... and I don't know if you have this. Did I give you this slide? So it's not, I'm not going to have you, do have it? Good, okay, I can't remember. So the way this works is that monoamine, tyramine is a monoamine, right? It's derived from tyrosine, and it itself gets converted to norepinephrine and epinephrine, okay? So that's a normal and healthy, good thing that happens. MAO is the enzyme that will degrade tyramine, and it will act on norepinephrine and epinephrine to detoxify them. So anytime you have a hormone, especially something as critical as a catecholamine, right, it's important that you have a pathway that breaks it down into a non-toxic metabolite. So if you have an MAO inhibitor, you're going to block that enzyme, and what happens is you get more of the tyramine that is around is going to get converted to norepinephrine and epinephrine. So you get a buildup of stress hormones, essentially. And that can lead to a hypertensive crisis, is what we call it, where you get wildly elevated blood pressure. You get heart palpitations, headache. And that's, so if somebody's on this medication, we tell them to avoid foods that are high in tyramine. So are there any diet techs in the room that may have done a diet or have been aware of low tyramine? What foods might you avoid? Anyone can help me out here? So sorry, say it louder. So really aged cheeses, right? Anything else? Everybody's talking at once and I'm deaf. Rachel, do you want to, I can. Fermented foods, yes. Any other? Soy, and particularly fermented soy foods, okay? Tempeh and I don't know what other ones there would be, but, and then anything else? What did I say, Sam? Cured meats. So aged and cured meats, aged cheeses, I heard Chianti, red wine, you know, and then there are probably some other ones. Oh, smoked fish. I didn't say smoked foods, right? We said aged and cured meats, but smoked meats also tend to be high in tyramine. Tofu, miso, those are other soy foods. Tempeh, sauerkraut. So fermented foods, just leave it at that. And there's a long list. If you ever have to give a diet instruction, you'll get a whole list for them. Okay, so about amino acids. We talked about how amino acids can be categorized by their R groups, that unique structure that you can't synthesize is how we define an essential amino acid. And then we can, you know, we've got all the various properties to think about. They can be large or branch chain, hydrophobic, hydrophilic, sulfur-containing, hydroxyl group containing, in fact. Second way is by their essentiality. I was getting ahead of myself. So remember what essential means is you can't make that structure. The R group is particularly important. Essential means you can't make it. We talked about conditionally essential amino acids, a few examples. And then the last way is by the fate of the carbon skeleton, which is important to keep in mind. And was that me? That wasn't me. Somebody dinged. Sorry, I'm going to just, I'm going between. So what happens to the carbon skeleton is another way that we think about and classify amino acids. So let me talk a little bit about that. So the first thing is, and actually let me back up just a moment to this. So let's put a definition over here by glucogenic. So what do you think glucogenic means? Take a stab. Anyone? Thank you, Dylan. Right, so the carbon skeleton can be used to make glucose. Now, you know the term anaplerotic? Have you heard that term, right? So that's what we mean by that, is that the carbon can add carbon to the TCA cycle and actually be used in gluconeogenesis. I'm going to show you this on the diagram on the next page. Ketogenic is and I'll just say this to make us move along here. The skeleton can be used to make fat, right? So all amino acid carbon can be used to make fat. Make sure you write this down, okay? So all of them can be used to make fat. Most, but not all, can be used to make glucose. So most of them are anaplerotic. But there's an important exception. And the one exception is shown here, and I know it's red on blue again, kind of hard. Circle leucine up here, because I don't think I have it animated or anything. Leucine's the only amino acid that cannot be used to make glucose. So it does not give rise to a net increase in carbon for the TCA cycle, whereas all the other ones do. So I'll show you leucine metabolism in more detail when we get to muscle, because that's an important tissue where leucine is used and fully oxidized. But for now, you can just think of it as it only yields 2 carbon end products, okay? So that's not giving rise to a net increase in carbon for the TCA cycle. So it is not glucogenic. It's only ketogenic. Okay, is that clear to everybody? The only one that can't be used to make glucose. No, the only one that cannot be, does not make glucose, or you can't use it to make glucose, is leucine. Yes, leucine up here. It's shown in red because it's the only one that is, it's uniquely, cannot be used to make glucose. It ends up in acetyl-CoA, 2 carbon units, right? So it's not giving rise to net increase. And that's it, I think. Anything in the summary that jumps out at you? I think I've covered all those pieces there. Incidentally, as you're going through the study guides after class, I hope you're doing that after class every time we talk. If there's something that I missed or it's unclear to you, please bring questions back, okay? But I'm going to kind of keep pressing forward here, so shout out if you have burning questions. Okay, so that's kind of the overview, and if you remember our roadmap, I can't remember, yeah, here's my roadmap here. Where we're going to be for the next rest of the time, pretty much, is here in the amino acid pool. And today we're going to focus a lot on what happens to the nitrogen that comes off when you break down an amino acid. We've just briefly mentioned what happens to the carbon skeletons. We're going to talk about that too. So what do you think urinary nitrogen, when we're excreting nitrogen from amino acid breakdown, what's the primary way that we do that? What is the primary form of nitrogen that we excrete under normal physiologic conditions? Urea, right? So we're going to talk a lot about the urea cycle, right? We already have introduced that. And I'm going to focus our discussion today and the next class period on specific tissues. So we're going to start with the liver, then we'll get to the intestine, muscle, and the kidneys, and then we'll talk a little bit about inter-organ glutamine metabolism, and that's kind of where I'm going to take us over the next couple of days here. All right, so some of this, hopefully, will be familiar to you because you've had biochem. And we're going to talk, and really we're going to stay in the fed state today, pretty much, when we're talking about most of what's happening at the liver. So after a meal, right, amino acids are coming in large volume through the portal vein to the liver, right? That's the main way that those are coming in. Under these conditions, we've got a large number of those amino acids that come into the liver are going to get catabolized immediately, almost. So the question that we're going to think about is, first of all, why are so many catabolized? So let's ask that first. Why is it that when you have a big influx of amino acids that come into the liver, why is it, do you think, that most of them get, or 60% or so, are going to be catabolized? Why is it that we do that? So if you take in a lot of glucose, what happens at the liver? Gets stored, doesn't it, as glycogen, right? So we don't store excess amino acids. We don't keep a depot of amino acids, right? We do protein synthesis. We'll make proteins as we need them, but anything in excess of that we use for energy, or we store it as fat, right? Because that's what we do. We just can't, there's no depot for it. So that's why. The how I'm going to talk about next. The where I want you to think about that the portal vein, right, It's going up into the liver, and surrounding that vein, the portal vein, are a specific set of cells called the periportal, P-E-R-I, I've written it somewhere in the sets of notes here. Periportal hepatocytes means liver cells that are right around the portal vein. So that makes sense, that a lot of what we're going to be talking about is happening very conveniently located as you're just entering the liver, pretty much, from the intestine. Okay, so primary main points about the liver. It is the primary site for uptake of amino acids after a meal. Okay, first pass metabolism. That's really important. Number two, it's the primary site for essential amino acid catabolism, except for one very important class of essential amino acids. And that is the branched chain amino acids. So all essential amino acids get catabolized pretty much at the liver. Not all of them, 60%, good many of them. The branched chain amino acids tend to be allowed to pass through unbothered by liver processes. And you're going to understand, if you don't already, by the end of this, why that's the case. Okay? And the third point about the liver is that it is regulating the availability of amino acids to the rest of the body. So it's this, again, this very important first pass metabolism. Regulates the availability of amino acids to the rest of the body. Okay, and I think here's my picture of, a truncated form, right? This is obviously not true to scale, but it does show, right, the hepatic portal vein. And this is the terminal ileum. Those of you that are MNT, I have this somewhere in MNT notes. I'll show you this at some point, probably the end of GI. And so obviously very truncated, right? But this is where bile salts are also reabsorbed. Vitamin B12 is absorbed down here. But the portal vein, very important, and the periportal hepatocytes are where we are going to be talking about a lot of the next few slides processes here. Okay, so the how. How are those essential amino acids catabolized? The first and most important amino acid reaction that I'm sure you've heard about, right? It's transamination. And this involves the transfer of an amino group from one amino acid to another, right? Just in very general terms. So here you got an amino acid unlabeled. We'll just do this in a general way at this moment. Now this is why I told you that glutamate is a key intracellular hub. It happens to participate in pretty much, well, I would say the vast, vast majority of transamination reactions are going to involve glutamate, right? So you take the amino group of one amino acid, you transfer it to the carbon skeleton of glutamate, which is alpha-ketoglutarate. And you've seen this, right? Where is alpha-ketoglutarate? Doesn't it show up somewhere? Do you call it TCA cycle? Yeah. Krebs cycle is the old, yeah, TCA cycle. Yeah, okay. So we have this around, right? It's an abundant entity. In so doing, you form glutamate, right? Because the amino group has now been transferred to the carbon skeleton of glutamate. And then you leave the alpha keto acid, which is, when I say carbon skeleton, that's a looser term for the alpha keto acid of that first amino acid, right? So there are a lot of different aminotransferases in the body, in the liver particularly. Most of them involve glutamate. Do you know what the two most active ones are? If you had to take a stab, have you learned this in biochem? I'm really trying to kind of feel out how much you've learned about. Do you have something? Can you say it? Is that Mackenzie? It's been a while. It's rusty. Okay. No one? So I've got them in the notes, actually, so you could look ahead if you wanted to. But we'll say, first one is aspartate transaminase, aspartate amino transaminase, I suppose, is probably the more, you'll see terminology shifting a bit. The other one is alanine, okay? So the next two slides we'll walk through this. Now, one thing you want to notice about transamination reactions, they are all reversible, okay? When I write it like this, I'm indicating it's a reversible reaction, can go either way. I've written it representing the directional flow that tends to predominate in the fed state. Okay, so in the fed state, we're producing glutamate from these transamination reactions. Now the other thing, I'm wondering, does anyone have any clue what, and I think I wrote it out, right, up here, pyridoxal phosphate. It's the active form of a vitamin. Anybody have any clue what that is? Yay, vitamin B6. Okay, where'd you learn that? Did you know it already, or did you learn it from one of our classes? What, you already knew it already? You're just, okay. Anybody else hear that in any other class? Huh? No? Nobody else? Maybe? It's all a blur, isn't it? It's like you're in senior year, you're ready to go. I don't think we do I don't know that we maybe teach you a lot about active forms of vitamins, okay? You need to know it now, though, okay? So PLP is pyridoxal phosphate, and that is the active form of vitamin B6. So vitamin B6 is hugely important in amino acid metabolism. And incidentally, vitamins and minerals are integrally involved in metabolism. And I think you have probably already, well, when we get to branched chain amino acid metabolism, I will highlight a few of the other vitamins that are involved, okay? But you know energy metabolism, right? Thiamine. Yeah, I'm looking at some, okay, so I'll show you some of the cofactors for, that are involved, and hopefully you'll have an appreciation for them. But B6, amino acid metabolism. So that was aspartate transaminase, and I'm assuming you're following here, right? Aspartate is the amino group gets transferred to alpha-ketoglutarate to make glutamate, leaving oxaloacetate, another TCA intermediate, right? And then alanine transfers its amino group to make glutamate, leaving pyruvate. So you can imagine how abundant these are, right? Because these intermediates are totally abundant. Okay, so points that I just want to make sure I made, right? Number one, these are always reversible reactions. Number two, in the fed state, we're moving in the direction of forming glutamate, right? I said that. Oh, I didn't say this. So number three is that these transamination reactions are a way of moving amino nitrogen around, right? You can, it's a way of making a non-essential amino acid from an essential amino acid, and it's also a way to make one non-essential from another non-essential. So we can interchange amino nitrogen around. And the other really important thing about these reactions, particularly at the liver, is that it is a way of feeding or transferring amino nitrogen as we're catabolizing amino acids, right? I said it's a lot of catabolism going on. It's a way of getting amino nitrogen into glutamate, and then from glutamate getting it into the ammonia that is feeding into the urea cycle. So we're going to talk about glutamate getting broken down and releasing ammonia that then goes into the urea cycle. So conveniently located in the periportal hepatocytes, okay? So that's what's happening. Lots of amino acids coming in. And we're going to be doing this to basically stoke the urea cycle as we are in the fed state. So I have a slide that I think will probably be a little redundant here. So why is it important? Right? It's a way of dealing with amino nitrogen to get it excreted into the urea cycle, right? Or getting it excreted as urea. And it's a way of equilibrating amino acid nitrogen so that we can have the amino acid pool as we need it to be. Which transaminases are most active altogether? The top two? Aspartate transaminase and alanine, okay? So knowing that, well, wait, and we're not quite there yet. Well, yes, knowing that, what are the three key amino acids involved in those two enzymatic reactions, AST and ALT? What are the three? Okay, louder, please. Or raise hand, maybe, so we don't all talk at once. Can somebody say? I heard mutterings. Okay, aspartate, right? How about anyone else? Come on, someone who hasn't spoken. What else? Please help me. Alanine, thank you. Glutamate. So now you are also in your mind thinking about the alpha keto acids that are involved, okay? You'll need to know these reactions, and if you know the enzyme, you know the players, right? This is not a memorization thing. This is just, right? Aspartate and AST and ALT. Which amino acid is the key intracellular hub? Glutamate, okay, great. I said they're reversible reactions. Which direction predominates? What are you forming in the fed state? Glutamate. So amino nitrogen is getting transferred, being pushed toward glutamate, and that's going to help us get amino nitrogen into the urea cycle. Which vitamin is required? Vitamin B6. What is the active form? PLP. All right. Yeah, we're doing rote learning now. I'm sorry. This is sort of repetition. Now, here's a new one. Do all amino acids undergo transamination as the first step of their degradation? No. Right, of course. When I ask a question like that, you know it's going to be, no, there are some exceptions, okay? The two exceptions are lysine and threonine, which is THR, if you remember the three-letter. Lysine, LYS, right? Lysine and threonine. Lysine is actually a very unique amino acid, not to be confused with leucine, which is unique for its own reason, right? The ketogenic amino acid, right? Don't confuse it. Lysine is unique in that it doesn't get easily degraded. It's very readily conserved, so that's a really useful thing. It's essential. Because it doesn't undergo transamination right away, it tends to be conserved a little bit. Now, if you look at the pathway and, go look up, you get metabolic charts, you'll see that there may be 8 or 10 steps down in its pathway. There might be a transamination reaction, but it's not the first thing that happens. So, just a little bit of a FYI. Another key point that I want you to remember is that transaminases have a high Km, which means low affinity for their substrate, right? So high Km means low affinity for their substrate. So that means that in the fed state, they're really going to be active because there's lots of amino acid around, right? So can you imagine why that's a useful thing? Yeah, so that means the liver's just not going to completely suck all the amino acids and keep it to itself, all the time. So as you're getting out of the fed state, as you're getting into post-absorptive state, that allows amino acids to go to the rest of the body and be used as is needed, right? So that's an important concept. Okay, second most important reaction for amino acids is deamination. It's an alternative way to remove the amino nitrogen group from some amino acids. Incidentally, this is just written up here as an FYI for the way I'm writing these reactions. And I think in most cases I was able to get in and modify these. I created these diagrams a long time ago when I taught metabolism at Virginia Tech. So I should be writing the free form as ammonium ion, okay? And that's because the pKa for this group is something like 9.5. So at physiologic pH around 7, it's going to be looking like this, okay? So if I ever write just NH3, just know that that's not exactly right, but it's representing the same thing. We're talking about ammonia, okay? And I'm going to forget, and I'm not going to say ammonium ion, okay? But it's ammonia, essentially. Amino group as it comes off. So this one is a dehydration, threonine dehydratase. So remember I just told you that threonine and lysine were the two that don't undergo transamination as the first step. Well, threonine undergoes a dehydratase step as its degradation. So it's not conserved like lysine is. It undergoes degradation, but just not by transamination. And it forms alpha-ketobutyrate, which is another one of those intermediates. Okay, next, right? So we're talking again in the fed state. We're pushing amino acid nitrogen into the formation of glutamate. Then what? So the next thing that happens is glutamate will get deaminated, right? So we move the amino nitrogen to glutamate, and then we deaminate glutamate to get that ammonia released to go into the urea cycle. So how does the glutamate get deaminated? There's an enzyme called glutamate dehydrogenase, and just like whenever you see dehydrogenases anywhere else, right? Where else have you seen, can you name a dehydrogenase that you're probably very, very familiar with? Pyruvate dehydrogenase, yeah, and there are other ones, right, that you've probably been exposed to. So you know that that uses a reducing equivalent, or produces a reducing equivalent, NADH, right? And that's useful, and at the same time, you know, releasing that ammonia, okay? Oops. And if you want to write it down here, you might as well. It's on the next slide as well. But glutamate dehydrogenase is a mitochondrial enzyme, okay? Now, before we move to the next thing, if you have a reducing equivalent and you're in the mitochondria, what do you suppose is going to happen to that NADH, or what could happen? Could it be used to generate something particularly in the mitochondria? Right, ATP, right, energy. So that's why I like to remember where things are happening, just because it helps me understand a little bit about what's happening after a reaction takes place. So this transamination followed by glutamate dehydrogenase has been termed transdeamination. When you're in the fed state and it's rapidly processing these amino acids, it's useful to think of it as a two-step process that's happening pretty much simultaneously, right? So transaminases, these are involved, they're everywhere. They're in the cytosol. They're also in the mitochondria. Glutamate dehydrogenase is only in the mitochondria. We are all completely, this is in the periportal hepatocytes. I've got to activate here. So in the mitochondria here, produces the reducing equivalent that provides much of the energy that's needed, and actually net cost of urea is very, very efficient. We only need one, it takes only one energy, but we'll walk through the energetics in a moment. Produces the ammonia that goes into the urea cycle. The first steps of that pathway are in the mitochondria as well. Okay, so it's a very efficient process. Any questions about anything so far? All right. Yeah, Dylan.
You said that transamination happens in the cytosol, deamination happens in the mitochondria.
Well, no, what I said is aminotransferases are everywhere. They're in the cytosol and the mitochondria. The glutamate dehydrogenase is in the mitochondria. So there are ways of getting, it's a little more controlled, but it's also producing the reducing equivalent that's used in large part, we can think of it, paper biochemistry anyway, right, because it's all happening all at once, but provides the energy needed that, for the urea cycle to be moving. So now that we've removed the amino group, this is the most exciting animation that's coming today. I'm just going to tell you, by the way, as soon as I click. I did look at this because I don't know what Bronwyn really likes doing this sort of thing. So here we go. Wah! Okay. Or just to wake you up. I don't know. So how do we get rid of the ammonia? That is first question on our agenda today. And the answer is we're going to make urea, right? You don't have this, but so just check out the structure of urea. Okay, it's got 2 amino groups. So we're going to talk about where those nitrogens come from, okay, as we go through the urea cycle here. And then after a while, we're going to talk about what happens to the carbon skeletons. But the first thing is we're just going to stay thinking about the urea cycle. So this is the urea synthetic pathway, and I say it that way because the first 2 steps are not actually technically part of the cycle. The cycle is really this part, okay? And I'm going to walk through it in a very methodical way, and then we'll come back to the big picture, okay? So the big picture is just so you can kind of gain an appreciation for or remember it, right? Did you learn this in biochem? Not briefly? Okay, well, now you're going to learn it a little bit more in depth. And sorry, I've got it animated. Okay, so we'll walk through it in the big picture, and then I'll do the breakouts, okay? So step 1 is up here, and I'm going to go through this where it's less busy in just a moment. Step 2 is this ornithine and carbamyl phosphate joining to make citrulline. Step 3 is citrulline getting converted to arginosuccinate. Step 4, arginosuccinate yields arginine and fumarate. And then the final step is you get urea formed and ornithine gets reformed, and that's why this is the cycle part of it, okay? And we're going to talk through all of these steps. Okay, the last step is the recycling of fumarate back to aspartate, okay? Let's break it down because it's a lot easier to follow this way. So, step 1. First thing to note is the enzyme, okay? And I want you to learn the enzyme names. Carbamyl phosphate synthetase is basically synthesizing carbamyl phosphate, right? So if you want, you can abbreviate this for ease, just so you can recognize it. But when you see the abbreviation, I want you to say in your head carbamyl phosphate synthetase so that you know what the enzyme's doing, okay? So step 1, wait, I'm sorry, my animations are always... The first thing to note is that at this step, you're taking ammonia and bicarbonate, which is CO2, essentially. That's the way bicarb looks in the body. You're cleaving 2 energy equivalents. So it takes 2 energy equivalents to synthesize carbamyl phosphate. And you've got these important cofactors up here. So NAG, which really sounds terrible, is N-acetyl glutamate. It is an allosteric activator of that enzyme. So if you don't have N-acetyl glutamate, you don't have CPS1 activity. Okay? So this is a cofactor. You got the concept of cofactors, right? Magnesium is also a cofactor. Every time you see ATP being involved in a reaction, you're going to see magnesium, and it stabilizes the interaction of ATP with the enzyme. Okay? N-acetyl glutamate, again, absolutely required for this CPS1 to function. Now, the first thing I want you to note is that the first nitrogen group of urea, remembering this structure, comes directly from ammonia. All right? So, woo, I almost spilled the drink. That would have been really not good. So let's talk about where is ammonia coming from, okay? So this is a little bit of a list. Where do you think it's coming from? Do you have some idea? We've just talked a little bit about where it might be coming from. Amino nitrogen, right? And directly coming in from glutamate, right? So it's this amino acid nitrogen for sure. As we're catabolizing those, transaminating them, moving them to glutamate, that's where it's coming from. We also have, so we're talking about the portal vein, right? So we have all those amino acids coming in, they're getting transaminated, moving it to glutamate, right? We also have other ammonia coming in from the portal vein, actually, from the gut. And I think I told you at the beginning something about urea, whatever. You can have ammonia being released from bacterial working on urea that gets into the bloodstream, and then that will also come in to the liver. Probably more likely is breakdown of bacterial proteins, right, that will release some ammonia. We get DNA and RNA also that get broken down from various cells and cellular components in the gut. So nitrogenous compounds that we talked about, DNA and RNA, purines and pyrimidines. You remember those are the nitrogen-containing components that will yield ammonia. We also get ammonia from foods we eat. Some of those more fermented foods actually have some ammonia as well. And then gut metabolism, intestine is going to be it's a big consumer of glutamine. And we're going to talk about the intestine. But glutamine metabolism just in the intestine yields a lot of ammonia. So there's ammonia coming in from other sources. A lot of it's amino acid in origin, but some of it's from food and some of it's from bacterial working on various compounds. Okay, and so that's the first nitrogen. So that's one of the questions we'll ask at the end. That was the step one. Step two, and by the way, we're in the mitochondria still. Remember how we said that, glutamate dehydrogenase and the first two steps of the pathway are in mitochondria. Second step is OTC, ornithine transcarbamylase, is the enzyme that basically takes ornithine, combines it with carbamyl phosphate to make citrulline. I forgot to tell you that I have these little boxes with the 5C. I'm not giving you the structures because it's really messy if I try to do this. Plus it's really small when you try to show you structures. So I'm just trying to help you think about it. 5-carbon ornithine combines with this 1-carbon carbamyl phosphate, very simple compound, to make citrulline. And that enzyme OTC is catalyzing that reaction. That's number 2. All right. Then what we have is we need a carrier, right? A transporter that will bring citrulline out of the mitochondria into the cytosol. And I'm going to tell you at the end, and over here we've got ornithine coming into the mitochondria on the same carrier. So that's a convenient thing. Ornithine and citrulline use the same transporter to come in and out, and they do it in opposite, they're exchanged for each other. Katarina. You're going to learn a lot about ornithine, so hold that thought. It is an abundant compound, but we can make it from amino acids and other things. And do I see a hand in the back or not? No, that's just stretching. Sorry. Okay. So step number 3, we've got this citrulline, 6-carbon compound, now combines with aspartate, right? One of our important amino acids. It's a non-essential, but it's an important one. And the enzyme that catalyzes it is called arginosuccinate synthetase. So again, I wouldn't recommend abbreviating this. Just say it out loud. It's easy enough to spell because you're making arginosuccinate from these 2 compounds. And this is another place where we cleave the equivalent of 2 ATPs. So we've now used the equivalent of 4 ATPs, right? We're down 4. The other thing you should just, if you want to write out here, that this is considered the rate limiting step in this cycle. And it's also, it's also where, we're getting the other nitrogen that is in urea is coming directly from aspartate. Okay? So aspartate is losing its amino group to, coming into arginosuccinate. And at the end, we'll talk about how we end up with urea, okay? So I'm not showing you the structures. That's the disservice of not showing you structures, is that it's harder to envision this. But all right. Step 4 is you cleave arginosuccinate to arginine and fumarate, right? So now we've just generated arginine, the amino acid, non-essential, but very important. And how many nitrogens does arginine have? Yes, good. 4 nitrogens. It's got a lot of nitrogens, which is convenient. Wait, was there anything else I wanted to say about that? No, I guess not. And then we got the last step. Step 5 actually produces urea and ornithine, right? So arginine gets cleaved to yield urea and ornithine. Ornithine is going to be coming into the mitochondria as that citrulline is coming in. Again, paper biochemistry, but it's happening simultaneously. Uses the same transporter, so it's convenient. So ornithine is getting recycled, so that's why we call it a cycle, right? So we can regenerate the entities to keep it going in a really rapid way when we're in the fed state and we've got lots of amino nitrogen to deal with. The other thing I want to show you, okay, so this is not on your slide, but just remember what arginine, I showed you the structure at the beginning of our time together, so that's huge, and I'm going to move it again, but this is what comes off. That's urea. Okay, so 2 nitrogens, first one coming from ammonia, second nitrogen coming from aspartate, right? Now the last step is this, that we have to, so we've recycled ornithine, we now need to recycle fumarate to get it back to aspartate so we can keep the cycle going, okay? So fumarate, actually, sorry, I'll keep animating that. Fumarate gets converted to malate. We got malate dehydrogenase, which you've heard of before, making OAA. Then we've got OAA and glutamate with the enzyme, the transamination reaction, AST, yielding aspartate and alpha-ketoglutarate. So I've written this. This is essentially the AST reaction minus the PLP and other things, okay? So is that clear to everybody how we can recycle fumarate back to aspartate? When this malate dehydrogenase goes, we produce an NADH. I had told you, we're making NADH, the reducing equivalents from glutamate dehydrogenase too, so there's plenty of reducing equivalent around. This one is in the cytosol, right? So it's a little bit of a loose association here that ultimately, because you produce NADH and you've got the shuttle, right, that gets it into the mitochondria, essentially the net cost of urea synthesis is 1 ATP, right? Because we've cleaved 4 ATPs, but we produce an NADH that ultimately yields 3 ATP. So the net cost is very efficient. It's very efficient to do this, and that's good news because we've got typically way more amino acid nitrogen coming into most of our diets than we need, and we need to be able to get rid of it without causing ammonia toxicity, right? I mean, essentially the urea cycle is pretty critical. So let's look at the energy dynamics again. I got that up there, I guess. If I said it, any questions that I have not highlighted, I'm not sure, but here's the cycle again. So first step, you're cleaving 2 ATP. Second is this rate-limiting arginosuccinate synthetase. Then in the recycling of fumarate, you produce a reducing equivalent that gains you 3 ATP, and that's how you explain the net cost is 1. Okay? So it does behoove you to you're probably going to have to memorize this cycle, but once you do, it'll be an easy thing to answer questions about it. Okay? So the idea is that net cost is pretty low for urea synthesis. Are there any questions at this juncture? And you're going to have to tell me when we need to stop. We don't stop. Oh, we should stop soon? Oh, okay. Well, wait a minute. Wait a minute. Hold everything. Let's, can we finish the urea cycle and then take a break? Would that be okay? Okay. I've got all these things. What else? What else just animated? I can't even see it. Ah. Yeah, net energy cost. Okay, for those of you that hate cycles, here it is written out in a linear way, okay, step by step for you. And I think I got everything. It's messy no matter how you look at it, but hopefully this will help you too, okay? And I think it's all true to form, so I won't go over it again, but maybe as you're learning it. Okay, let's go over just these key points about the urea cycle again. And I have to remind myself, where am I? So the first step, or first question is, where does the carbon come from? So what do you think? Where does the carbon come from? So look, yeah. No, so let's go back. So yeah, and what is bicarbonate, really? It's CO2, right? You've been paying attention to metabolism, and you know that we produce CO2, don't we, right? So CO2 is produced from metabolism of various nutrients. So we have it in the, it's around. It's an abundant source. So the first carbon in the urea cycle comes from CO2. And if you go back and look at the cycle, you'll see it's bicarbonate, right? So you know that's where it is. And then you got where do the two nitrogens come from? As the second question here, where do the two nitrogens come from? So one is from aspartate, and the other is ammonia, right? And ammonia coming from lots of different places, but it's being produced. How much energy does it cost? Oh, you know, this is one. Okay, one. So you're going to be able to say, yep, first step, CPS1 cleaves 2. Another 2 equivalents are being cleaved, used at the arginosuccinate synthetase step. And then we produce an NADH in the recycling of fumarate, and the net cost is 1. So that's the complete answer there. Just so you just kind of can talk through it and think about it. Where does ureagenesis occur? First of all, where in the body, where are we happening? What's this big picture? Where are we? Where did we start today? No, we didn't start there. We're in the liver, right? We're in the liver. And then what particular cells in the liver? Where are we? Periportal hepatocytes, right? And then where in the cell? Well, right? First 2 steps are in the mitochondria, and the rest of the cycle is in the cytosol of cells, of these periportal hepatocytes. And then the last major points is that we're recycling fumarate and ornithine, right? And so that's why I broke it down for you, so you can be really clear about why it is, or how it is that we're recycling these things, right? So we start with ornithine, we end with ornithine. We make fumarate, and then we recycle it, right, back to aspartate. And what's the importance of that, right? How would you explain why that's important? Please. Well, that I'm not thinking about how exactly that might happen. Why would that happen, right? So if you didn't recycle them, they might build up, I guess, right? What else? What would be the immediate consequence? Yeah, Rachel? The cycle would not be a cycle, would it? Right, it would stop. Yeah, so, right, you could have, those things could build up, but they'd stop, and then we wouldn't build up, and we wouldn't make them anymore. We would just stop. We wouldn't make urea, right? I mean, it's just, so you've got to be able to, any cycle, right, recycle the substrates to keep it going. The how does that happen, I'm not going to go back over that, but I want you to know that, okay? So, just think about how the ornithine, gets recycled and how fumarate does. What regulates the urea cycle is a fairly, maybe we should stop there. Let me give you a break. We'll come back and talk about what regulates the urea cycle. I can't see quite the clock. Should we say quarter till to come back? Or is that good? Oh, thank you. That's so much closer. I can see that. What do we give you guys? What do they get? 8 minutes. Oh, what are the chances we're going to be able to do that very well? Okay, 8 minutes.